Hello students, let's talk about amino acids today. Amino acids are organic compounds containing amine and carboxylic acid as their functional groups and a side chain specific for each amino acid. They are also the building blocks or monomeric units of proteins. Besides being the constituent of proteins, amino acids have varied functions as diverse as neurotransmission and biosynthesis of porphyrins, purines, pyrimidines, and urea. Out of over 300 naturally occurring amino acids, 20 constitutes the monomeric unit of proteins. Amino acids are often designated by either a 3-letter abbreviation or a 1-letter symbol. The abbreviation of amino acid are the first 3 letters of their names, except for asparagine, glutamine, isoleucine, and tryptophan. The symbols for amino acids are the first letter of their names. The other symbols are agreed upon by convention. Structure of amino acids. All 20 of the common amino acids are alpha amino acids. They have a carboxyl group and an amino group bonded to the same carbon atom, the alpha carbon atom. They differ from each other in their side chains or the R groups, which vary in structure, size, and electric charge, and which influence the solubility of the amino acid in water. Since the alpha carbon in amino acid is a chiral center having bonded to 4 different groups, all amino acids except glycine can exist in 2 stereoisomeric forms, L and D stereoisomers. D amino acids are found in very few peptides, including small peptides in bacterial cell wall and certain peptide antibiotics. Classification of amino acids. Amino acids are classified into 5 main classes based on their R groups, particularly their polarity. The polarity of the R group varies from highly nonpolar to polar. The first class, nonpolar aliphatic R groups. The R groups in this class are nonpolar and hydrophobic. The amino acid in this class includes glycine, alanine, valine, leucine, isoleucine, proline, and methionine. Glycine has the simplest structure with a hydrogen atom as the R group. Alanine, valine, leucine, and isoleucine are highly hydrophobic side chains that normally cluster together within the protein structure with hydrophobic interactions. Second class, aromatic R groups. The amino acid phenylalanine, thyroxine, and tryptophan have aromatic side chain as their R groups, and they are relatively nonpolar, that is, hydrophobic. All the 3 can participate in hydrophobic interactions. They can also absorb ultraviolet lights, and it accounts for the characteristic strong absorbance of light by most proteins at a wavelength of 280 nanometer. Thyroxine and tryptophan are significantly more polar than phenylalanine because of the presence of thyroxine hydroxyl group and the nitrogen of the tryptophan indole ring. The third class, polar uncharged R groups. This class of amino acids includes serine, threonine, cysteine, asparagine, and glutamine. They have R groups which are more soluble in water or more hydrophilic. This is because the R groups contain functional groups that form hydrogen bonds with water. The polarity of serine and threonine is attributed to their hydroxyl groups, that of cysteine by its sulfahydryl group, and that of asparagine and glutamine by their amide groups. Asparagine and glutamine are the amides of 2 other amino acids also found in proteins, aspartate and glutamate, respectively, to which asparagine and glutamine are easily hydrolyzed by acid or base. Cysteine is readily oxidized to form a covalently linked dimeric amino acid called cysteine, in which two cysteine molecules or residues are joined by a disulfide bond. Cysteines are highly hydrophobic and plays a significant role in protein structures. Fourth class of amino acid, positively charged basic R groups. This class of amino acids includes lysine, arginine, and histidine. The R group in this amino acid have significant positive charge at pH 7 and they are highly hydrophilic. Lysine has a second primary amino group at the epsilon position on its aliphatic chain. Arginine has a positively charged guanidine group and histidine has an imidazole group. The fifth class of amino acid, negatively charged acidic R groups. This class of amino acid includes aspartate and glutamate, each of which has a second carboxyl group. These two amino acids have net negative charge at pH 7. Uncommon amino acids. In addition to the 20 amino acids, proteins may contain residues created by modification of common residues already incorporated into the polypeptide. Among these uncommon amino acids, 4-hydroxyproline, 5-hydroxylysine, 6N-methyllysine, carboxyglutamate, selenocysteine, etc. Amino acids have both acid and base properties. When an amino acid is dissolved in water, it exists in solution as a dipolar ion, where the carboxyl group loses a proton and the amino group gains one. Such dipolar ions with no net charges are termed as deuterions. Deuterions can act as either an acid, which is a proton donor, or a base, a proton acceptor. Substances having this dual nature are amphoteric and are often called ampholytes from amphoteric electrolytes. A simple monoamino monocarboxylic amino acid such as alanine is a diprotic acid when fully protonated. It has two groups, the carboxyl group and the amino group, that can yield protons. The characteristic pH at which the net electric charge on an amino acid is 0 is called the isoelectric point or isoelectric pH, designated as Pi. The solubility and melting points of amino acids reflects their ionic characters. The charged functional groups of amino acids ensures that they are readily solvated by and thus soluble in polar solvents such as water and ethanol, but insoluble in nonpolar solvents such as benzene, hexane, or ether. Similarly, the high amount of energy required to disrupt the ionic forces that stabilizes the crystal lattice accounts for the high melting points of amino acids, which is greater than 200 degrees Celsius. Amino acids do not absorb visible light and thus are colorless. However, as mentioned earlier, thyroxine, phenylalanine, and especially tryptophan absorbs ultraviolet light, which is in the range of 250 to 290 nanometer. Functional groups detects the chemical reactions of amino acids. Each functional group in an amino acid exhibits all of its characteristics, chemical reactions. For carboxylic acid groups, these reactions includes the formation of esters, amides, and acid anhydrides. for amino groups, acylation, amidation, and esterification, and for hydroxyl and sulfhydryl groups, oxidation and esterification. The most important reaction of amino acid is the formation of a peptide bond. Peptides are chains of amino acids. Two amino acid molecules can covalently join through a substituted amide linkage termed as peptide bond to yield a dipeptide. Such a linkage is formed by removal of the element of water, dehydration, from the alpha carboxyl group of one amino acid and the alpha amino group of another. Although peptides are written as if a single bond link the alpha carboxyl and the alpha nitrogen atoms, this bond in fact exhibits partial double bond character. Therefore, there is no freedom of rotation about the bond that connects the carbonyl carbon and the nitrogen of a peptide bond. Three amino acids can be joined by two peptide bonds to form a tripeptide. Similarly, amino acids can be linked to form tetrapeptides, pentapeptides, and so forth. When a few amino acids are joined in this fashion, the structure is called an oligopeptide. When many amino acids are joined, the product is called a polypeptide. Proteins may have thousands of amino acids residues. Although the term protein and polypeptide are sometimes used interchangeably, molecules referred to polypeptides generally have molecular weights below 10,000, and those called proteins have higher molecular weights. An amino acid unit in a peptide is often called a residue, the part left over after losing a hydrogen atom from its amino group and the hydroxyl moiety from its carboxyl group. In a peptide, the amino acid residue at the end with the free alpha amino group is the amino terminal or N-terminal residues. The residue at the other end, which has a free carboxyl group, is the carboxyl terminal or the C-terminal residue. The peptide bond is uncharged at any pH of physiologic interest. Peptides, nevertheless, are charged at physiologic pH owing to their carboxyl and amino terminal groups, and where present these acidic or basic R groups. As for amino acids, the net charge on a peptide depends on the pH of its environment and on the pK value of its dissociating groups. I would like to conclude this topic with these few points. Out of about 300 amino acids found in nature, only 20 amino acids constitute the building blocks of protein, one of the most abundant macromolecule in nature. The 20 amino acids are only L stereoisomers with a configuration related to the absolute configuration of the reference molecule L glyceraldehyde are found in proteins. The chemical nature of the R group in amino acid gives this characteristic property like solubility, chemical reactions, etc. All amino acids possesses at least two weakly acidic functional groups, amino and carboxyl group. Many also possesses additional weakly acidic functional group such as hydroxyl, sulfhydryl, guanidino or imidazole groups. Amino acids can be joined covalently through peptide bonds to form peptides and proteins. The primary structure of a peptide is its amino acid sequence starting from the amino terminal residue. Proteins can be very long polypeptide chains of 100 to several thousands of amino acid residues. However, some naturally occurring peptides have only a few amino acid residues.